Today, we can map the entire genetic profile of a novel virus in a matter of hours, or program engineered bacteria to manufacture human insulin. But our fluency in this biological code is remarkably recent. For decades, scientists knew that physical traits were passed down between generations, but they had no clear understanding of the physical mechanism responsible for heredity. In 1928, Frederick Griffith discovered a non-lethal bacterial strain could acquire lethal traits from its remains. A physical substance transferred disease instructions. Griffith called this the transforming principle, proving heredity was driven by a tangible substance, DNA. This discovery left biologists in a difficult position. They had identified the molecule that stored life's data, but they lacked the technology required to read, decipher, or edit the sequences written on it. Identifying DNA was only the starting line. The real challenge lay in understanding how different life forms physically architect and organize these massive amounts of data. By the 1970s, scientists developed techniques to manually cut and paste genetic material together, an achievement known as recombinant DNA technology. But as researchers began splicing genes, they found that simple and complex organisms store their information using very different structural rules. Bacteria, or prokaryotes, are built for high efficiency. Their genetic information is held in a single circular chromosome, floating freely in the cytoplasm. Here, genes are organized into operons, continuous strings of coding DNA grouped together without wasted space. This streamlined, continuous structure made bacterial genomes an ideal proving ground for early genetic engineering, as the code was easy to locate and relatively simple to manipulate. Complex life forms like plants and humans utilize a far more guarded architecture. Our DNA is linear, massive in scale, and sequestered inside the cell nucleus. Because of its length, this DNA must be spooled around histone proteins, forming a condensed material called chromatin. This packaging allows 2 meters of human DNA to fit into a space just micrometers wide. Unlike the compact code of bacteria, eukaryotic genomes are interrupted by introns, non-coding sequences that must be edited out before the data can be used to build proteins. In the early days of sequencing, this complexity was an insurmountable wall. At the speed of manual laboratory methods, reading a 3 billion-letter human genome would have required centuries of continuous effort. Early techniques, such as Sanger sequencing, were limited to reading very short fragments of DNA at a time, making them poorly suited for analyzing large, complex genomes. The solution arrived in the mid-1990s with high-throughput shotgun sequencing. This method shatters the genome into thousands of random fragments, reads them simultaneously, and uses algorithms to stitch the overlapping pieces back together. This shift to computational analysis led to the complete sequencing of a bacterium in 1995, followed quickly by yeast, fruit flies, and eventually the entire human genome. High-throughput technology finally provided the tools necessary to parse these massive datasets, regardless of how complex the underlying biological architecture was. The ability to sequence whole genomes provided a new vantage point, adding a holistic perspective to a field that had traditionally relied on a purely reductive focus. In the past, researchers isolated a single gene to see how it produced a single protein. This provided deep detail, but only for a fraction of the system. Today, we track how thousands of genes and proteins interact across an entire cell at once. This perspective has given rise to fields like transcriptomics and proteomics, which analyze global biochemical networks to see how a living system functions as a unified whole. By deciphering both the streamlined code of bacteria and the dense archives of human cells, we have moved from observers of individual traits to become modern researchers who are now fully capable of mapping, comparing, and actively modifying the massive molecular systems that drive life.